Hello, everyone. This is Pocket Size Bio with Dr. Casco. And in this video, we're going to talk about chromosome mutations. In the previous one, we talked about point mutations in a single DNA sequence where you have substitutions, deletions, additions. In this video, we're going to talk about more the grand scope of chromosomes, right? And the types of mutations you can get from situations arising from chromosomes, right? So before we begin off, I just want to put into perspective, um, how chromosomes are essentially confined as, and especially with humans, that we create karyotypes, right? Um, so let's get to it. Genetic variations can also occur, right, from large changes to the chromosomes. This is like chunks of the chromosome going on and moving to a different chromosome, for example, right? And because of that, because of the nature of such a large part of a chromosome moving from one area to another, many genes are likely to be affected, right? Because a chromosome is a large portion carrying many genes, right? So many genes can be affected simultaneously, right? You could have a change in chromosome number entirely, depending on the species. That actually happens naturally, right? You could have some organisms that will become a haploid, or could be haploid individuals. You could have some that are polyploid, meaning that they have more than the one set of chromosome number, right? And depending on the cell type, you can even have changes in chromosome number also, right? What you see here in this panel is a karyotype, is basically a way that scientists have devised to kind of organize all the chromosomes based on similarities, right? Based on the size, the shape, where the particular bands are, right? And in our case of humans, we have this karyotype that shows the 23 chromosome pairs, homologous chromosome pairs, right? We have the 22 autosomes, and you have that 23rd sex chromosome pair, right? So I want to focus a little bit more now on variations of chromosome number. If you guys know, we are diploid individuals, right? Our cells are diploid with the exception of our gametes, right? And we basically have that 2n equals, 46 chromosomes um, in our nuclei, right? And as we undergo meiosis on our cells, we produce gametes that are haploid or haploidy, right? And when you have individuals that have half of the number of chromosomes, you refer to that as showing haploidy characteristics, right? Some individuals could normally be in this formation, right? And while others need to be in that diploid formation. So when you occur naturally as a haploid, right, you refer to that as being a monoploidy individual. And that occurs in colonial insects, right? Ants, bees, and wasps. And it's very, very interesting in the case of bees, right? Um, where you basically have females that are diploid and you have males that are monoploid. So that means they only have one chromosome set, not the two chromosome sets that we think about, right? Sorry. So in the case of bees, right, you have males, and males are referred to as drones in this case. So if you take a look at this diagram here, you have a male drone, which is haploid, and these little lines basically indicates the little chromosome set, right? Um, the DNA is expressed fully within, it has to be expressed fully because we only have one set, right? Um, and basically, male sperm produces by mitosis, not meiosis, but they still produce male sperm, right? Females, on the other hand, are diploid individuals where they produce their gametes by meiosis, right? To reach that haploid state. Okay. So take a look at this graph here. You have a queen, right? A queen bee that is diploid. Queens are diploid individuals, and they produce haploid eggs, right? They only have one chromosome set. Interestingly, if these eggs get fertilized, okay, by the haploid sperm of the drone, of the males, okay, basically, if we get this particular egg and this particular sperm, you would get a female. All right. Any egg that is not fertilized will automatically turn into a male bee. Okay. A male bee. And the same goes for those female um, diploid, egg laying workers, right? They would produce haploid eggs. But, social behavior of the drones, the men only fertilize with the queen, right? Um, all these will turn into male drones, basically coming from unfertilized eggs. But if you have a particular case where you have an egg that gets fertilized by a sperm and you apply to it a very nutritive diet solution that's referred to as a royal jelly, right? That will provide the nutrients and the compounds for these females to turn into, for this individual to turn into a new queen, right? So you have some hierarchy happening going around here, but ultimately, what I want you to emphasize on is that haploid individuals will basically turn into drones, males, and um, those individuals that are basically diploid, the females, but don't get this nutritive substance, will just turn into female workers. All right. So here's a case where you have monoploidy in the sense of the males being haploid. Okay. And then you have females that are being diploid there. All right. So this idea that an organism can arise just from a single egg cell, you see that in nature a lot. It's called parthenogenesis, right? And it's basically that process in which an organism can be formed from a single egg cell. And many organisms do that. It's quite common in algae and fungi. It's quite common. I don't want to say common, but it does appear in fish and reptiles and amphibians also, right? Where unfertilized individuals cells can become an individual. Okay. There is a downside to having monoploidy, right? Since you just have one set of chromosomes, you're much more susceptible to having deleterious mutations. And of course, since you have basically just one gene, one allele, right? If you get a deleterious mutation, um, um, that's pretty much game over there. You won't be able to have that particular gene, right? So diploidy is preferred in most species, just as a better chances of survival, right? So parthenogenesis. There are other forms of ploidy, right? So we talked about monoploidy. Polyploidy is when you have a cell that has more than the two sets of chromosomes, right? Being more than diploid. Um, basically, right? That's the meaning of polyploidy, right? So this can occur when cell divisions they fail, right? When haploid gametes productions fail, right? So gametes will, instead of being haploid, they're diploid. Okay. There's some mistake in the uh, meiosis, right? Where you have these divisions, right? You have that metaphase and you have that anaphase, and basically that telophase, um, where they're separating the chromosomes. There's an uneven distribution, right? So if you take a look, for example, here, right, where you have that first mitotic division where each kind of chromatid, one is supposed to go here, one is supposed to go here. Both of them ultimately go to one side of the cell division, right? So basically, you just have an empty egg there, right? That could happen. And then these two occur um, normally, for example, right? Um, and then instead of having that half the number, you'll have a individual or a zygote. Once fertilization occurs with the sperm, right? You have a zygote that can have 3 times the chromosome number, right? Instead of being diploid, it'll be triploid, for example, right? So this would be called a trisomic zygote. Let's say this egg cell fertilizes this empty egg cell, it'll be monosomic zygote. So you'll just have basically being monoploidy, right? The same thing can occur here. The same thing can occur here, right? So when you have 3N individuals, this is called triploidy, right? If you have 2 diploid gametes, let's say this happens in the egg, but it also happens in the sperm, right? And they fuse together, you would have tetraploidy. So you'll have 4 times the 4 of the chromosomes set, right? Homologous chromosomes, you'll have 4 of them, right? 4 of one homologous pair, tetraploid, right? Good. So this is referred to as polyploidy, right? Um, Not necessarily a bad thing, depending on the individual, depending on the species, right? Um, so the process of which a cell or an organism can get one or more complete extra set of chromosomes is called polyploidy, right? This is common in plants, right? Ferns, green algae, and a lot of flowering plants are polyploid. And this can increase the fruit size, right? And the amount of fruits being produced within the plants, right? It also helps in other properties like the hardiness of the fruit, right? There is a downside of being infertile, right? But it is good for farmers, right? Because they can produce many flowering plants, many fruits in one given go, right? Without necessarily having them to produce more and more and more and more. Um, so everyone can get the fruits and things like that, right? So it is good for farmers in terms of plants and produce, but definitely if it happens to humans, it is um, lethal. It is very lethal. It does happen. It's rare, but when it does happen, the likelihood of the survivalhood of the individual is quite low. Okay. And then you have aneuploidy, right? Where you have a condition, right? Where there is the addition of an addition or loss of one chromosome from a cell, right? So you have that to an individual, right? Let's say the 46 chromosomes, 23 pairs plus one single chromosome. Or you can have that you're missing one, right? And this is more frequent. This does happen more often than these polyploidy. Um, and uh, why this occurs is basically the homologous chromosomes, they fail to separate, right? Another word for separation is segregation, right? From each other, and instead they go into one cell. Okay. This is also known as non-disjunction. Okay. Non-disjunction. And it usually happens with one pair of homologous chromosomes. Um, and while the other, just one pair of homologous chromosomes, right? And the other ones pretty much stay the same. Each homologous pair goes to each side during meiosis and things like that, right? To give you an example, um, here you have this little um, panel here where you basically have that homologous pair. During meiosis one, each of them are supposed to go their own side of the cells, but they basically stay together, right? And then let's say um, they are able to kind of separate. In this case, where you would have each of them here instead of having just one, right? Red, red, blue, blue. You have 2 of them here. Okay. Doesn't necessarily happen in meiosis one. It could also happen in meiosis two, right? But when an organism having a non disjunction gamete, when it fuses with a normal gamete, let's say this is an egg here, and it fuses, sorry, this is a sperm and it fuses with an egg, you're going to have 3 times just that chromosome only. That's referred to as trisomy. Okay. And opposite to that, let's say you would have an egg or a sperm joining, fusing with a normal egg, you would just have monosomy, right? If a normal gamete fuses with a gamete that has no chromosome, it is called monosomy, right? And this is the case that happens with individuals with Down syndrome, right? Where they have trisomy 21, right? They have three chromosome a set of three chromosomes for the chromosome set 21, right? We're going to take a look at there. So you have non-disjunction. Some other characteristics, sorry. Um, uh, some examples of these abnormalities that would occur, right, is Turner syndrome, which Turner syndrome is basically a monosomy in the sex chromosomes, right? So you would have an individual that will look something like this, right? Short stature, low hairline, um, certain characteristic facial features, right? Generally, um, um, poor development, right? They'll be infertile individuals, shorter than normal, right? Um, you would have these individuals with something called Turner syndrome, right? So they would either be XO because they don't have the other chromosome, or YO, right? They're missing that chromosome pair. All right. And then you have another individual case called Klinefelter's syndrome. Here, in this case, you have trisomy um, in the sex chromosomes. Now, here, you would have individuals that are XXY, right? Where they will generally be tall in stature, right? Frontal baldness would be absent, slightly feminine physique, right? They're phenotypically men, but the reproductive organs are pretty much underdeveloped, so they'll be infertile, right? They could be receive treatment of testosterone, but you would have these conditions here, right? This would be um, trisomy in the sex chromosomes. And then you have Down syndrome, where you have trisomy in the chromosome pair 21, which is an autosomal condition, right? And this could vary in symptoms, right? They generally have almond-shaped eyes, as you can see here in this little infant, right? Round face, shortened body parts, right? Generally heart defects, but they could develop, right? So these are some examples of chromosomal abnormalities in humans, right? Okay. And lastly, I want to talk about variations in chromosome structure, right? So we talked about mutations in the chromosome level, and there are additional things that can occur in terms of the chromosome structure in itself, right? This is different from crossing over. These are situations that could happen, breaks, right? When you have those double strand breaks, right? Changes in chromosome structure due to double-stranded breaks, right? Occurs during meiosis, okay? And is promoted by mutagens, right? And we discussed mutagens in the previous chapter, in the previous video, right? And it is possible to be repaired, right? Because DNA and the molecular mechanisms of DNA could um, has a proofreading mechanism, but there are times, again, like I have mentioned before, that it is difficult to be restored, right? So what happens in these examples, right? There's several types. You have deletions, right? Inversions, translocations, duplications, right? So in the case of deletions, that's just pretty straightforward, right? You have a break occurring in the DNA at two locations, right? Let's say between C and D and F and G, right? And that entire portion of the chromosome got removed, right? And then the resulting segments get fused together, right? Big chunk of DNA gets removed out, usually fatal, right? There's some conditions that can occur called Williams syndrome. The likelihood of survival is quite low, right? Um, you have inversions where you have a similar situation where you have a break in the middle, right? That middle piece gets removed out, it flips over, right? rotates 180 degrees and re-enters back. Okay. This is referred to as an inversion. All right. Not as dramatic, could lead to fusion of genes, right? Depending on what genes you're actually are um, being succumbed to this situation, right? You will, of course, have a misalignment in meiosis, okay? Because during meiosis, they align well, the alleles together. But because this is a type of variation in chromosome structure, you have this mutation, there could be a misalignment in meiosis, right? Um, Likelihood this happens that happens is reduced fertility, right? Translocation is quite interesting. Translocation is basically where you get a chunk of one chromosome, let's say half a chromosome, breaks in half, and it attaches itself to another chromosome, right? So here you have a break in this little red chromosome here, number one, cuts in half, and then that smaller portion, that lower portion, gets connected to another chromosome. Okay. Um, and depending on where it is, what genes are being influenced, it is involved in cancer formation, right? Referred to as translocations. And this one basically stays as a short stature there of chromosome. And then you have duplications where you're basically repeating, right? You're copying a segment of a chromosome, right? Many times, it could happen that multiple copies are being made, right? Um, so you have this duplication. In this case, you have FGHI, and you have that original chromosome, but now you have an extra set of that chromosome section that it gets attached to the same chromosome, right? It does have some advantages, right? Um, but generally harmful, right? Where you have these repeats of genes and chromosome section. Okay. Um, in the Waves program, you don't get into so much detail other than this, right? So don't worry too much on critically on how it actually works. Okay. Um, yeah. So that essentially wraps up 4.6 on the different types of mutations, right? Point mutations, chromosomal mutations, and ploidy. And um, the last thing I want to talk about is essentially how sexual reproduction and this idea of passing of genes, right, increases variation. In the next chapter, we're going to talk about evolution and inheritance and how genes can influence passing of genes, um, enhances variation within a population of species, right? But we've talked about uh, several points of variation when we discussed meiosis, right? There's several ways that you can enhance variation just by sexual reproduction, right? And the first one, if you remember, crossing over, right, where you have the homologous chromosomes. Let me just zoom in here. You have homologous chromosomes that they exchange little chromosomal arms between the um, non sister chromatids, right? To provide variation among the alleles of a particular gene there, right? So that's one way you can enhance variation. This is also known as recombinant chromosomes, right? Another way is basically the law of random segregation, right? Where basically um, the combination when you're creating this these gametes, right? We don't know just because of random assortment, right? Which allele you'll get between a homologous um, uh, chromosome pair. So if you see here, you have these two homologous chromosome pairs, right? One, two. And then when they undergo meiosis, you have these two little alleles, right? Both being blue, 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 blue. And then you have your other set, right? Red, red, A and B. But at the same time, just because of just a random assortment between all these chromosomes, you can get different combinations, right? So you can get chromosome number one, for example, this large A and this small B, right? You can get variations there. So that's completely random. There's no way that you can control that, right? So that's one other way that you can kind of add to a layer of variation within sexual reproduction, right? Crossing over, random assortment, okay? And ultimately, fertilization, right? Fertilization is your last form of variation because okay, you've created these random combinations. You've created within the chromosomes, you've created recombinant chromosomes, because of crossing them over. And then lastly would be how do you know which sperm will fertilize the egg? You know, each the sperms produce and the eggs have their own variation of recombination and random assortment. And then on top of that, you have um, a random fertilization. So these three things can provide a good deal of variation in a population of species, right? So it's quite interesting. We're going to get into in chapter 5 how these inheritance patterns work and the probability of getting a certain outcome. Okay. With that, we finish chapter 4 on variations and mutations. Okay.